Hello all, my name is Ryan Abdelaziz and I am pleased to have you guys join me for my video discussion. I'll be answering the question or I will be responding to the prompt of the reproducibility crisis in biotechnology. I'm going to go through this kind of sequentially, starting with the first question, which is the crisis in biotechnology, is the reproducibility crisis in biotechnology primarily a design problem, culture problem, or incentive problem? So I think primarily it's an incentive issue and operationally it's a design issue. So I think what that means is the overarching issue is incentives, and that incentives kind of drive what is researched, how it's researched, what results you look for, further studies based upon that research. And then within the individual studies, we kind of see the design problems resulting from the overarching incentives being, why are you looking for that research? And because of that, you might have some design issue. So I kind of want to discuss this in the realm of neuroscience because that is where I currently work and what I studied. So in neuroscience, there is an issue of statistical analysis. We often use like different types of modeling and different levels of analysis to sort of model pathways in the brain and find significant, structures in the brain and what they innervate and things like that. The issue is that the power of these analysis depends on sample size, effect size, and variance between brains. And often in neuroscience, we have very small sample sizes, and we also have quite a bit of variance. Like we are using statistics to detect very subtle signals and subtle changes in the brain, which is a very noisy and high dimensional system, and it's very, very complex. So I believe that it can be a bit difficult to attribute statistical findings as true in neuroscience. I also think that reproducibility is kind of an issue in neuroscience because before even any analysis happens, you have to standardize the tissue that you're collecting from, and it's quite hard to standardize tissue across labs. I can give you an example. So in one of my research projects, we are working on schizophrenia, and we have to take caudate. Caudate is like a very widely defined region. You can take, head of caudate, body of caudate, but you can also take caudate from broader striatal areas, and all of these samples within this region are slightly different, which can lead to downstream molecular data that is not from the same biological substrate. So that's kind of a reproducibility issue within neuroscience. And then we can use statistics to sort of account for these differences in the actual biological substrate. But I think that introduces further issues down the line. So the reason I say that all of the incentive issue is the broader issue is because I think funding is what drives research. And I think funding can kind of determine what is researched, how marketable that research is, how that research is designed, what methods are employed, and what results you look for. And that's why I kind of think that the incentive of why you do the research is kind of the larger issue, and then you have downstream design issues that result from that. Okay, for prompt 2, should reproducibility be universal or can a single finding warrant publication? So I believe that a single finding can warrant publication so long as it's framed as hypothesis generating and not as a standalone true observable statement. So these single observation or single studies should include transparent methods. It should have limitations on the study, and there should be enough detail in the study for it to be tested or refuted. Tested, accepted, or refuted, sorry. And stronger claims should require much stronger evidence. So claims that are significant to biotechnology, as in therapeutic or diagnostic or clinical claims, need much, much higher reproducibility and a much higher amount of evidence. There's a much larger burden of evidence there. But I do think single findings can warrant publication. I think there's some use in that, in having a single finding, because that generates further research into a single observation. Third question is, what single shift would most improve reproducibility. So I'm going to go back to the funding issue because I believe that funding is the overarching thing that drives incentive, which drives research, which drives experimental design flaws. So I believe that if funding was reformed so that so that research comes before funding, and that you don't seek funding to perform research, but rather you are just funded as a researcher, I believe that would fix quite a few issues. Like I previously mentioned, funding kind of drives what research is marketable. It kind of drives, it has like some bias towards what your results will be. And I think it's a bit difficult to have an unbiased, completely flawless research study without changing the funding of how or without changing how that study is funded. So yeah, I've gone a bit over time, but thank you all for listening. I'm eager to see what you guys think of my response, and to respond to you guys as well. So thank you.